Protomap hypothesis. A major challenge to students of the cerebral cortex is how individual and species-specific cytoarchitectonic areas have emerged from the initially seemingly uniform ventricular zone and cortical plate. Both intrinsic and extrinsic factors have been suggested. One attractive model, known as the tabula rasa hypothesis, is that all cortical neurons are equipotent and that laminar and areal differences are induced by extrinsic influences exerted via thalamic afferents. Creutzfeldt, 1977. And Creutzfeldt is spelled the same as Creutzfeldt-Jakob. However, there was also considerable evidence that the cells generated within the embryonic cerebral wall contained some intrinsic information about their prospective species-specific cortical organization. The protomap hypothesis was proposed as an empirical generalization in an attempt to reconcile available experimental and descriptive data in the field. Rakesh, 1988. It was based on the fact that all ontogenetic columns are mixtures of several clones and are not equal in each prospective cytoarchitectonic area. The protomap model suggests that the basic pattern of cytoarchitectonic areas emerges through synergistic, independent interactions between developmental programs intrinsic to cortical neurons and extrinsic signals supplied by specific inputs from subcortical structures. According to this hypothesis, neurons in the embryonic cortical plate, indeed even in the proliferative ventricular zone where they originate, set up a primordial map that preferentially attracts appropriate afferents and has a capacity to respond to this input in a species-specific manner. The prefix proto was introduced to emphasize the primordial, provisionary, and essentially malleable character of the map, which is subject to considerable modification by extrinsic influences exerted at later stages. The initial indication that developmental events in the proliferative ventricular zone foreshadow prospective regional differences in the overlying cerebral mantle comes from the observation that the neurogenesis of the primary visual cortex, which contains more neurons per radial unit than the adjacent areas, lasts longer. Furthermore, it has been demonstrated that the mitotic index in the ventricular region subjacent to this area is higher than in adjacent areas. In addition, initial establishment of cytoarchitectural and functional features specific to the visual cortex, such as the ocular dominance columns, form independently of thalamic inputs to the area. Therefore, certain region-specific differences in the production of the ventricular zone can be detected even before neurons arrive at the cortex. In addition, several lines of evidence indicate that during the final cell division, one or both daughter cells start to express a variety of neuron class-specific signaling molecules. Postmitotic cells not only become committed to a neuronal fate, but also become restricted in their repertoire of possible fates. Numerous studies in which the cytology of postmitotic cells has been examined and or manipulated by a variety of methods such as spontaneous mutations, ionizing radiation, retroviral gene transfer labeling, transgenic inserts, and heterochronic transplantations all indicate that certain class-specific cell attributes are expressed before the migrating neurons arrive at the cortical plate and become synaptically connected. Remarkably, neurotransmitter secretion is unnecessary for proper brain formation. In addition, retroviral tracing experiments and some clonal analyses suggest that the ventricular zone is comprised of a heterogeneous population of cells and that the cell lineage contributes substantially to the cell fate determination of neurons. Emerging observations confirm and extend these findings both within the cortex, in migrating interneurons, and later postnatally. These findings raise the question of whether laminar and areal identities of cortical plate cells provide cues or chemotactic attractants for incoming afferent axons. Data from axonal tracing indicate that afferent connections from the subcortical structures and other cortical regions find their way to the specific regions of the cortical plate either directly or by way of the subplate zone or both, suggesting the existence of region-specific attractants for pathfinding and target recognition. In support of this idea, the development of correct topological connections in anophthalmic mice and in enucleated animals indicates that basic connections and chemoarchitectonic characteristics can form in the absence of information from the periphery. The gradients or region-specific distribution of various morphoregulatory molecules, or both, in the embryonic cerebral wall, or layer-specific expression of POU, homeodomain genes, may also contribute to the formation of specified axonal pathways. For example, electroporation of FGF8 applied before beginning of formation of thalamic connections has shown that morphoregulatory molecules can shift the anterior-posterior areal boundaries in the developing neocortex. Thus, tangentially and radially distinct landmarks in the postmitotic cells facilitate axonal pathfinding and target recognition that eventually lead to parcellation of the cerebral cortex. It should be underscored that although the embryonic cerebral wall exhibits gradients of several morphoregulatory molecules as well as other specific area-specific molecular differences, the protomap within the embryonic cerebrum provides only a set of species-specific genetic instructions and biological constraints. The precise position of interareal borders, the overall size of each cytoarchitectonic area, and the details of their cellular and synaptic characteristics in the adult cerebral cortex are achieved through a cascade of reciprocal interactions between cortical neurons and the cues they receive from afferents arriving from a variety of extracortical sources. Such afferents may serve to coordinate and adjust the ratio of various cell classes with the subcortical structures, as has been shown in the primary visual system. Those references are all in the 90s. In summary, the concept of the cortical protomap includes the role of both intrinsic and extrinsic determinants in shaping the final pattern and relative sizes of the cytoarchitectonic areas.